populations. But once populations divide, or as they admix and as they're exposed to different environments, these different elements can have different histories. And usually there are some that have dramatically different histories, and they don't reflect sort of the average history of the genome overall. Skin color markers are one example. I mean, they are very different between populations, very different within continental regions too. It's not as if you're walking across the world and suddenly people change to a different color, or suddenly there's an obvious shade difference. But it's totally continuous and clinal, and often relates to latitude, because the sun has been an important factor in determining how skin color has evolved. For that reason, there's this extra level of difference in the genes that underlie skin color as a trait. Some people have also argued that, well, skin color is the only trait that determines race. So there's only 6 race genes. And I've seen this in several editorials, and this chain of logic. But I mean, it's really quite simplistic, because skin color isn't really determining race. I mean, an albino African person is still an African person.
To study these different components of what define race.
Race is, I mean, it's really many things, actually too many things to, kind of like God. I mean, if it's all these different things, how can you talk about it as just one thing? And this is my problem, and many people's aversion to using the word, because it's just all-encompassing. Or at least it it includes major social factors as well as biological factors.
I think most people are to some extent caught up in the sort of dilemma of what exactly does it mean to use the term race. And there has been, I think, a sort of tendency to use superficial indicators of race for lack of availability of better ones.
Some of the risks of these old ways of thinking, or just immature ways of thinking about human variation, are people not getting past their racialized perspectives. On a more scientific level, we're unable to really observe reality as well as we could if we had a better model, better general framework. So I think there's a lot of risks to society and to the individuals within society by enforcing, categorical definitions of human variation.
We all have common genetic variants that occur in the ballpark of every few hundred bases. So if you have 3 billion base pairs of DNA, between every 200 or 300 and every 1,000 various parts of the genome, there will be a specific base sequence that is different from one individual to another. These are the so-called single nucleotide polymorphisms. So, if you count that up, there are going to be probably in the range of millions to tens of millions of these variants. Now, as a percentage of the whole genome, the usual quote is that we're 99.9+% identical, but of course that 0.01% or whatever it is begins to add up when you have 3 billion base pairs. So probably on average in the millions to maybe 10 million or so differences from individual to individual. And some of those are going to be things that are relatively stable variants in different parts of the world, but will be different from one population to another.
We are one species, and one species that has a very recent common origin. If you compare human diversity with diversity of whales, for example, it's hugely different. I mean, whales go way back, and they've spread throughout the oceans, as we have throughout the world, but we've only spread throughout the world in a very recent time frame. If you compare us to other apes, you find again that the diversity is between 2 and 5 times greater in apes. So relative to other species, we're very much the same. Relative to the general capacities, humans are very much the same. I think physiology has been a very successful field of research because humans are very much the same. We don't really have to account for genetic variation at every step that we're investigating how the human body works. At most loci, there's not really a big difference between populations. And at some loci, there's no difference between populations. But at some other loci, there's very big differences.
This question of will we benefit is a question that goes to whether it's in part a risk question, whether the risks of participating in genetic research will outweigh any potential benefit. And why do I ask these questions? First, because the history isn't good. And that is, historically, genetic information has not very good genetic information, but it was believed to be good at the time. Genetic information has historically always been used in ways that adversely affected minorities, African Americans and other groups. It has always been used to show inferiority. It has been used to make some groups seem superior to others. That's just the way, the social utilization of the information. The second point is genetic information is very powerful information. And that is, we believe that genetics in this society, that genetics is predictive. And we know not a great deal at all about the interaction between our genes and our environments, for example. We don't know very much about the interaction between genes and genes, as well as genes and environment. Nonetheless, we have thought of genetic information as very deterministic. So that if you have a negative association with a piece of genetic information, we have tended as a society to push that in the direction of saying something harmful or stigmatizing about a population. So with respect to our ethical issues, we are working against a tough history. And part of the effort has to be, can it be turned around? Can we use genetic information today in a way that's beneficial or positive rather than in a way that's negative?
I come from a health perspective, a health prevention perspective. So then it doesn't, because I don't care if you're white or black. All I care is that you have diabetes. So I want to know why you have diabetes. Or I want to know if you're going to have a higher probability of having diabetes. I want to know which one of those blacks and white genes you have that might contribute for the disease. So it doesn't matter your race. It matters everything else. I will never have a study where I'm going to put admixture alone. It's too simplistic. It's me believing that only genes will have an influence. Sometimes what people do is that they run the statistical model and they add SES, social economical status, and SES was not significant. I'm going to take it out. That's something that I don't think is appropriate, because I think that even though might not be significant, it plays a role in models. When I see those kind of things, if I'm a reviewer, for example, I immediately make a comment about that, But the majority of people, what they do is that they just split people in categories, and that's what they use, and that's what we've been doing forever and ever. So, I think that judging that is right or wrong, it's I think that by the time it's been accepted so far, now I'd rather say that there are better ways to do it, and that we all should be challenged and encouraged to do it in a better way.
What we could call your genealogical ancestry, or who are the people you know were your ancestors. Like say your grandparents. If one of your grandparents was Native American, the other three were European, then you would be 1/4 Native American, because one out of the four was Native American. I would call that your genealogical ancestry. That could differ, even dramatically so, from your genomic ancestry, what we measure on you. Even if, that was a 100% Native American grandmother and the other three were 100% European. The process of the segregation of alleles that come down to you would make your result vary. And some people with that same genealogical ancestry could have 11% Native American ancestry. Other people might have 45% Native American ancestry. Which of those is true? I mean, do you really have 25% ancestry that's Native American, or do you have 11%? It kind of depends on what the question is. Is the question, well, what's my genealogical ancestry, or what's my genomic ancestry? And maybe in a legal sense, you really care about the former question, genealogical ancestry. Maybe biomedically, the latter, what's your actualized or your genomic ancestry, might be a more important answer. So, I mean, even in that very simple case, that person might self-identify, yeah, I'm a 1/4 Native American. He would be measured as something different. So that right there's going to be a variability in, between what they actually have that you would measure with genetic markers and how they think of themselves.
We define admixture as a composite value that reflects your ancestral information based on a finite number of markers. And I make that very, very clear. Because if I take your blood sample right now and I mention 10 markers, okay, those 10 markers can tell me, okay, you are 70% European-like, 20% African-like, and 10% American Indian. What if I measure 50 markers? That 70% can go to 65 or can go to 75. Because you see, the accuracy of that value depends on how much information you have. So the more markers we have, the more precise the information is. You see, and that's something that I make very clear. The way we define it, it is based on a finite number of markers. And we identify these markers based on frequency. If I have marker A, and I have two forms, yellow and blue, if yellow is more frequent in European populations and blue is more frequent in African populations, then I call that marker an ancestry marker, which is an ancestry informative marker, or AIM. So I take marker 1 and I check what you are. If you're yellow, yellow, you're European, European. If you're blue, blue, you're African, African. If you're yellow, blue, you're European and African. So then if I have 10 markers, different markers like that, then I can add up and create a composite value. That is not that simple as that. No, it's not. It requires more complicated. It requires statistical methodology. We use a lot of Bayesian approaches to, it depends on the information of the sample. But that gives people the idea of how this value comes into the picture.
There's been a study in the relatively recent past of a particular combination of medications for treating heart failure. The investigators used basically self-identification of African American ancestry as the kind of indicator for individuals who might respond better to this form of treatment than others. I don't fundamentally believe that they think that that is the best indicator. I think they would probably, if you were to ask them, acknowledge that it's really just a kind of loose surrogate for other things. But what I guess happens is, because it's the easiest way to go and it's the one that's most likely to be easily translatable into some clinical activity, that's what gets used. And so whether it was intended or not, it's the message that gets communicated that somehow so-called race matters in terms of, in this case, making decisions about medication. I think most investigators, if you were to, if it's relevant to the research, would quickly acknowledge that it's a very poor indicator. We wish we had better, but that it tends to be what gets used because it's easiest.
There may be some markers that make certain drugs work better or worse for different people, and these markers, and many of these have been studied. Some examples are P450 phase 2 metabolizing enzymes. So there's a whole series of defenses that our body has against exogenous chemicals. And these have adapted to be different in different parts of the world because there's different plant species largely that we're exposed to in different areas of the world. So there are some of these differences that clearly suggest pharmacogenetic differences might exist in how people are responding to drugs. But if your groupings are really so large and heterogeneous, the likelihood of anybody in one particular group having all of the variants that may be even typical for that group, maybe the average person has those, is very small. In African Americans especially, there's a lot of admixture. I mean, there's some people that have more African ancestry and other people that have more European ancestry and others that have more Native American ancestry within this large group that you would call black or African American. So knowing or projecting a drug that would treat them, especially if it's a drug that is also then different in how other populations can utilize the drug or respond to it, is very tenuous. And I think if there are real differences that make the drug better for people with African ancestry generally, there's going to be some people in that sample that this drug is not better for, and they may be more European at those few loci.
I think what will probably come of this at some point in time is, if there is a difference that's reproducible between self-identified African Americans and others as it relates to a particular medication, to me that means that there are probably a gene or maybe several genes that are represented at higher frequency in African Americans than others on average, and which are responsible for that difference. And the real challenge would be to find those genes, because in an ideal world, what you would do isn't to test for admixture, and it's not to test or just to use self-identification. It's to say that the reason, on average, African Americans respond better to this drug than somebody else is there's a greater probability that the following 1 or 2 or 3, whatever number it is, genetic variants are going to be found as greater in African Americans than somebody else. And what you'd really like to do is test for those specific things. Then you could do it in anybody. But I think we're still a ways away, and I view it as a stepping stone in a sense. The fact that self-identified race can show that there are differences in drug response is an important first step towards figuring out what the genes are that are responsible for that, and then eventually, from a clinical point of view, developing an approach to looking at those particular genes.
And really that's where everything is headed. So I think we're sort of at an intermediate point. Again, we have to think about snapshots in time. So here, this is kind of the second point in what I see as a progression from understanding that there are epidemiological differences, to then quantifying those and seeing if they really are there, and if they're biological, or if they're cultural, and how to understand those two things. And then moving on to finding the actual genes that determine those differences. And using those in a clinical situation is generally what we call personalized medicine, and it's kind of where people think we're headed with some of these diseases.